Part 2. Lipids as signaling infrastructure. Chapter 5. Lipids as information carriers. Information within the cell is not limited to genetic sequences or soluble signaling molecules. It is embedded in the composition and organization of lipids that define membranes. Each membrane encodes a set of physical conditions that determine how proteins behave, how signals propagate, and how the cell interprets its environment. This encoding is not symbolic. It is mechanical. Change the lipid composition, and the behavior of receptors, channels, and enzymes shifts because the environment they operate in has been altered. Lipids therefore act as carriers of information by setting the constraints under which cellular processes occur. The bilayer itself functions as a continuous input surface. Fatty acid chain length, degree of saturation, and head group composition determine packing density, curvature, and permeability. These properties influence how proteins insert into the membrane, how they move, and how they interact with one another. A membrane enriched in tightly packed lipids creates a different signaling landscape than one with greater disorder. Receptors embedded in these environments adopt different conformations and respond differently to the same ligand. The information is not in the ligand alone. It is in the structural context that defines how that ligand is processed. Lipid diversity allows the cell to generate localized environments with distinct properties. Different regions of the same membrane can support different behaviors by varying lipid composition. This creates zones where specific processes are favored, such as signal initiation, transport, or mechanical stabilization. Proteins that require certain conditions localized to these regions, effectively reading the lipid environment as a set of instructions. When lipid composition changes, these instructions change, and proteins redistribute or alter their activity accordingly. Signal transduction often begins with lipid modification. Enzymes alter specific lipids in response to external input, changing their structure and, in turn, the properties of the membrane. These changes can recruit proteins, alter membrane curvature, or modify permeability. The signal is carried through the membrane as a change in its physical state, not just as a cascade of soluble intermediates. This allows the cell to integrate information across a surface rather than relying solely on diffusion through the cytosol. Time is encoded through lipid turnover. Membranes are continuously remodeled, with new lipids incorporated and existing ones modified or replaced. The rate and direction of this turnover determine how long a given state persists. Rapid turnover allows quick adaptation to changing conditions, while slower turnover stabilizes existing configurations. This creates a temporal dimension where the history of inputs influences current behavior through accumulated changes in membrane composition. Lipid-mediated signaling extends beyond the cell surface. Internal membranes, including those of organelles, carry their own lipid compositions that define their function. Communication between compartments depends on how these membranes interact, fuse, or exchange material. The compatibility of lipid environments influences whether these interactions occur efficiently. Differences in composition act as barriers or gateways, controlling the flow of information within the cell. Disturbance of lipid composition alters the information landscape. Changes in packing, curvature, or domain formation shift how signals are initiated and processed. The same external input can produce different outcomes because the membrane no longer presents the same structural conditions. This does not require changes in receptor number or ligand concentration. It arises from changes in the medium through which signaling occurs. Lipids carry information by defining the architecture of the system. They determine how components are arranged, how they interact, and how signals are transmitted and resolved. The cell reads this information continuously, adjusting behavior based on the structural state of its membranes. Understanding signaling, therefore, requires understanding how lipid composition encodes and modifies the conditions under which all cellular processes operate. Fatty acids as signaling precursors. Fatty acids serve as stored signaling potential that can be released, transformed, and redeployed in response to changes at the membrane. They are not passive structural elements once incorporated into phospholipids. They are substrates held in reserve within the bilayer, positioned for rapid conversion into signaling molecules when enzymes act on specific sites. The membrane therefore functions as both a barrier and a reservoir, where the composition of fatty acids determines what signals can be generated and how quickly they can be produced. Release begins with enzymatic cleavage. Phospholipids target defined positions on membrane phospholipids, liberating fatty acids that were previously constrained within the bilayer. The identity of the released fatty acid determines the signaling pathways that follow. Arachidonic acid, for example, enters a sequence of reactions that produce eicosanoids, which influence vascular tone, immune activity, and cellular stress responses. Other fatty acids follow different metabolic routes, producing signals with distinct effects. The membrane composition, therefore, sets the range of possible outputs before any stimulus occurs. Conversion pathways define how these precursors are used. Once released, fatty acids are rapidly metabolized by enzymes that transform them into active signaling molecules. These transformations change the structure of the molecule, altering its interaction with receptors and its ability to diffuse through the cellular environment. The rate of conversion and the balance between competing pathways determine which signals dominate. If one pathway is favored, its products accumulate and shape the cellular response. If another pathway becomes more active, the profile of signals shifts accordingly. Spatial control is maintained throughout this process. Enzymes responsible for release and conversion are localized to specific regions of the membrane, ensuring that signaling occurs in defined areas. This localization allows the cell to generate signals where they are needed rather than uniformly across the membrane. The proximity of enzymes to their substrates reduces delay and increases the precision of the response. Changes in membrane organization can disrupt this arrangement, altering where and how signals are produced. The physical properties of fatty acids influence their signaling behavior. Chain length and degree of saturation affect how these molecules interact with enzymes and receptors. More unsaturated fatty acids are more flexible and can adopt conformations that facilitate interaction with a broader range of proteins. More saturated fatty acids are more rigid, influencing how they are processed and how they integrate into signaling pathways. These structural differences translate into functional differences in how signals are generated and how long they persist. Signal termination depends on further metabolism and reintegration. Once fatty acid-derived signals have exerted their effects, they are broken down or re-esterified into membrane lipids. This restores the reservoir and resets the system for future signaling events. The efficiency of this process determines how quickly the system can return to baseline. If termination is delayed, signals persist and overlap with new inputs, increasing noise. If termination is efficient, signals remain discrete and controlled. The balance of fatty acids within the membrane determines the baseline signaling capacity of the cell. A membrane enriched in specific fatty acids will produce a different set of signals than one with a different composition. This balance is not static. It changes over time as new fatty acids are incorporated and existing ones are modified. These changes alter the signaling potential of the system, influencing how the cell responds to future stimuli. Fatty acids act as signaling precursors by linking membrane composition to dynamic cellular responses. The cell does not generate signals from nothing. It draws from a pre-existing pool embedded within its structure. The nature of that pool determines the range, intensity, and duration of signaling events, making membrane composition a direct determinant of how information is generated and processed. Eicosanoids and local control systems. Eicosanoids are generated at the site of membrane disturbance and act within a constrained radius, shaping conditions locally rather than broadcasting signals across the entire system. Their production begins when specific fatty acids are released from membrane phospholipids and immediately enter enzymatic pathways embedded in the same or adjacent membrane regions. This tight coupling between release and conversion limits diffusion and ensures that the signal reflects the conditions at that exact location. The result is a control system that operates through localized adjustments in vascular tone, permeability, immune activity, and cellular behavior, all determined by the lipid composition present at the time of activation. The pathways that produce eicosanoids compete for the same substrates. Once a fatty acid such as arachidonic acid is liberated, it can be directed into cyclooxygenase, lipoxygenase, or related enzymatic routes. Each pathway generates a distinct set of molecules with different effects on surrounding cells. The balance between these pathways determines whether the local environment shifts toward constriction or dilation, activation or resolution, recruitment or containment. This balance is not decided after the fact. It is set by enzyme availability, membrane organization, and the relative concentrations of precursor fatty acids embedded in the bilayer. Localization defines both the strength and duration of the signal. Eicosanoids act on nearby receptors and are rapidly degraded or converted into less active forms, preventing widespread propagation. This rapid turnover allows the system to adjust conditions in real time without creating persistent systemic effects. When production is controlled and termination is efficient, the local environment can shift quickly and then return to baseline. If production exceeds termination capacity, signals persist longer than intended, extending their influence beyond the original site and altering neighboring regions. Membrane composition determines what signals can be produced and how they behave once formed. Different fatty acids yield different classes of eicosanoids, each with specific effects on cellular function. The proportion of these fatty acids within the membrane sets the baseline signaling profile. A shift in composition changes the distribution of signals generated during activation, altering how the local control system responds to the same stimulus. This does not require changes in receptor expression or enzyme levels. It arises from the available substrates embedded within the membrane. Receptor interaction translates these signals into action. Eicosanoids bind to specific receptors on nearby cells, initiating changes in ion flux, enzyme activity, and gene expression. These effects alter vascular diameter, immune cell behavior, and tissue permeability. Because the signals are produced and act within a confined space, the response remains targeted. Cells outside this region remain unaffected unless the signal persists long enough or spreads far enough to reach them. Structural integrity of the membrane influences the precision of this system. When lipid organization supports controlled release and conversion, signals are generated where needed and terminated when their function is complete. When membrane structure is unstable, release becomes less regulated, and conversion pathways may shift unpredictably. This produces overlapping signals with competing effects, increasing variability in the local response. The system remains active, but it loses clarity and control. Eicosanoids function as a local control layer embedded within the membrane. They translate structural conditions into immediate adjustments in the surrounding environment, allowing the cell and its neighbors to respond to changes without relying on distant signaling systems. Their behavior reflects the composition and organization of the membrane because that structure determines both the inputs and the pathways available for signal generation. Structural origin of inflammatory signaling. Inflammatory signaling begins as a consequence of altered membrane structure rather than as an isolated biochemical decision. The membrane defines baseline conditions for containment, separation, and controlled exchange. When those conditions shift, either through mechanical disruption, oxidative modification, or changes in lipid composition, the system detects a loss of structural integrity and initiates a response. This response is not triggered by abstract signaling intent. It emerges from changes in packing density, permeability, and domain organization that alter how proteins behave and how molecules move across the boundary. Disruption of lipid packing is the first event that changes signaling behavior. Tightly packed membranes restrict movement and maintain separation between compartments. When packing loosens, transient spaces within the bilayer increase, allowing ions and small molecules to pass in ways that were previously constrained. This alters local concentrations and electrical conditions, which directly affects membrane-bound proteins. Channels open under different thresholds, receptors adopt different conformations, and enzymes encounter substrates at rates that deviate from baseline. These changes create the initial conditions that favor activation of inflammatory pathways. Fatty acid composition determines the type of signals that follow. Membranes enriched with certain fatty acids provide substrates that can be rapidly converted into signaling molecules when released. Enzymatic cleavage of these lipids produces precursors that enter pathways generating eicosanoids and related compounds. The identity and proportion of these fatty acids set the profile of signals that will be produced. A shift in composition alters the balance between pathways, changing whether the local environment favors recruitment of immune cells, alteration of vascular tone, or resolution of the response. Reactive byproducts reinforce structural disruption. When electron leakage or other processes generate reactive species, these molecules interact with membrane lipids, modifying their structure and further reducing packing stability. Oxidized lipids change how the bilayer behaves, increasing permeability and altering domain formation. This creates a feedback loop where initial disruption leads to chemical modification, which then amplifies the structural changes that initiated the response. The membrane becomes both the source and the target of the signaling process. Protein localization changes as structure degrades. Receptors and enzymes that rely on ordered domains for proper positioning become dispersed or misaligned. This alters how they interact, increasing the likelihood of unintended activation. Proteins that are normally separated may come into contact, initiating pathways that would not be active under stable conditions. At the same time, proteins that require clustering for proper function may fail to assemble, weakening coordinated responses. The result is a mixture of activation and dysfunction driven by loss of spatial control. Mechanical stress contributes to this process by altering membrane tension. Stretching or deformation changes how lipids pack and how proteins embedded within the membrane behave. Mechanosensitive channels respond to these changes by altering ion flow, which further modifies intracellular conditions. This links physical forces directly to biochemical signaling, integrating structural disruption with cellular response. The membrane translates mechanical input into chemical output through changes in its own organization. Signal propagation depends on whether the structural changes are contained or spread. Localized disruption produces localized signaling, allowing targeted responses that can be resolved once the structure is restored. Widespread disruption extends these signals across larger regions, increasing the scale and duration of the response. The difference lies in how stable the surrounding membrane remains and whether it can prevent the spread of altered conditions. Resolution requires restoration of structure. Lipid repair, replacement, and reorganization return the membrane to a state that supports controlled exchange and stable protein localization. As packing density increases and permeability decreases, the conditions that supported inflammatory signaling are removed. Signals diminish not because they are actively suppressed in isolation, but because the structural environment that allowed them to persist no longer exists. Inflammatory signaling originates from changes in the material state of the membrane. It reflects how the system responds when the boundary that defines cellular organization is altered. The specific pathways that activate are determined by the available substrates and enzymes, but the initiation of the process is structural. Restore the structure, and the signaling resolves as a consequence of returning the system to a stable operating condition. Signal timing and termination. Signal timing is set by how quickly components assemble into a functional complex and how long that complex is held in place by the membrane. Initiation begins when receptors encounter ligands within a domain that supports clustering. The rate of that encounter is determined by lateral mobility and local density. When the membrane allows controlled movement, receptors find one another and assemble within a defined window, producing a signal with a sharp onset. If mobility is excessive, components fail to remain in contact long enough to stabilize the active state, delaying or weakening initiation. If mobility is too restricted, encounters occur slowly, stretching the onset over time and reducing responsiveness to changing input. Propagation depends on maintaining proximity across sequential steps. Kinases, adapters, and downstream effectors must bind in order, each interaction occurring before the previous one dissipates. The membrane enforces this sequence by confining these components within domains where interaction rates are high. When domains are intact, the signal moves forward with minimal delay because each step occurs within a localized environment. When domains fragment, components disperse between steps, increasing the time required for the next interaction and raising the chance that the signal will decay before completion. Timing becomes variable because the structure no longer holds the sequence together. Amplitude and duration are linked through retention. A cluster that persists allows repeated interactions, reinforcing the signal and extending its duration. This persistence is not indefinite. It depends on the balance between forces that stabilize the cluster and processes that drive disassembly. Lipid composition sets this balance by determining how tightly components are packed and how easily they can separate. Stable packing supports sustained interaction long enough for full signal transmission. Instability shortens retention, truncating the signal before downstream pathways are fully engaged. Termination requires coordinated disassembly and deactivation within the same spatial context where activation occurred. Phosphatases, lipid-modifying enzymes, and receptor internalization machinery must access the active complex while it is still localized. The membrane facilitates this by positioning termination components near activation sites. When this arrangement holds, signals end cleanly. Receptors return to baseline, and downstream molecules are reset. If activation and termination are separated spatially due to domain disruption, deactivation lags behind initiation. Signals persist beyond their intended duration, overlapping with new inputs and increasing noise. Chemical gradients contribute to timing by controlling how quickly signaling intermediates are removed or converted. Molecules generated at the membrane must either diffuse away, be degraded, or be reincorporated into the membrane. The rate of these processes determines how long the signal remains active. Efficient clearance shortens duration and sharpens resolution. Slower clearance extends duration and increases the likelihood of interaction with unrelated pathways. Membrane permeability and domain integrity influence these rates by controlling diffusion paths and access to enzymes. Electrical conditions also shape timing. Changes in membrane potential alter the behavior of voltage-sensitive proteins, shifting activation thresholds and affecting how long channels remain open. Stable potential supports predictable timing, while fluctuations introduce variability in both onset and termination. Ion fluxes that respond to these changes feed back into the system, influencing enzyme activity and further modifying signal duration. Signal timing and termination are therefore structural outcomes. The membrane defines how quickly components assemble, how long they remain together, and how efficiently they are dismantled. When structure supports controlled assembly and coordinated disassembly, signals begin and end with precision. When structure degrades, initiation becomes delayed or inconsistent, and termination becomes incomplete or prolonged. The system continues to process input, but without clear boundaries in time, leading to overlapping signals and reduced clarity in output.